Hello students, welcome to the biology class. In our session we will discuss about glycolysis. In our previous sessions we discussed about the types of respiration, in which we explained aerobic and anaerobic respiration, and the mechanism of respiration. The glycolysis is considered to be the common step for both the aerobic and anaerobic respiration. Whether the respiration takes place in the presence of oxygen, that is your aerobic respiration, or in the absence of oxygen, that is anaerobic respiration, the first and the common step for both the aerobic and anaerobic respiration is glycolysis. What do you mean by glycolysis? Glycolysis is the process in which the glucose is broken down into the two molecules of pyruvaric acid, that is known as your 3-carbon compound. So from 6-carbon compound you will get 2 carbon compounds, that is your pyruvaric acid. So what is that process known as? So glycolysis is the breakdown of glucose into the pyruvaric acid, that is your 3 molecular compound. The another name of glycolysis is EMP pathway, because this systematic formation or the 10 steps of glycolysis is given by Embedden, Meyerhoff, Paranis. So that is EMP pathway. So because of that, that pathway is also known as your EMP pathway. Where it takes place? It takes place in the cytoplasm. So the site of glycolysis is the cytoplasm of the cell. So the first step you can see, the glucose, it undergoes the glucose 6-phosphate. Means the addition of the phosphate has taken place in the presence of enzyme hexokinase. From where that phosphate has come, that is from your ADP is converted into the ATP. Means here we have utilized the energy in the form of a phosphate. So here we have -1, in which we have utilized the energy. The second is glucose to fructose. Structurally they are different, but molecular formula is same. So we simply say isomerase. The glucose is converted into fructose, that is known as the isomerase. Now fructose 6-phosphate forms the fructose 1,6-diphosphate, or you can say 1,6-biphosphate. Again the addition of phosphate is there in the presence of enzyme phosphofructokinase with the utilization of energy, that is ATP. Again we consume the ATP. Here again we have consumed the ATP. So the first step and the third steps of the glycolysis are those steps in which we are consuming, means utilizing the energy in the form of a phosphate. Now fructose 1,6-diphosphate is converted into 2 compounds, that is 3 carbon and 3 carbon, that is dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. Now this 3-phosphoglyceraldehyde and dihydroxyacetone, they are irreversible to each other. This 3-phosphoglyceraldehyde will undergoes the dehydrogenation and they will form 1,3-biphosphogyceraldehyde. Again there is the addition of phosphate. So here there is no phosphate, one more phosphate is being utilized. So we are having ATP to ADP. Fine. So there is the reduction is there, NAD to form the NADH, because the reduction is there, and here we have taken the coenzyme, that is NAD, which is being converted into NADH2. Now this biphosphate is converted into 3-phosphoglyceraldehyde. Means here one phosphate is synthesized. So that is ADP to ATP, and here we have gain 2 ATPs. Fine. Now 3-phosphate to 2-phosphoglyceraldehyde. This 2-phosphoglyceraldehyde means simple, the change of the position of the phosphate is there, from 2 it is on 3. So that is called as the mutase. Then 3-phosphoglyceraldehyde is converted into 2-phosphoenol pyruvic acid, that is enolase, and the removal of water is there. Now this 2-phosphoenol pyruvic acid is converted into the pyruvic acid, which is the end product of your glycolysis. That is, in the presence of enzyme pyruvate kinase, again we have synthesized the energy, that is in the form of 2 ATPs. ADP is converted into ATP. यहाँ का phosphate भी हमने utilize कर लिया, यहाँ का phosphate भी utilize किया. 4 ATPs are there, but we have consumed 2 ATPs, that is your -1 and -1 is there. So 2 minus 4. So ultimately the gain of ATP is only 2 ATP directly. But here we can have NADH, which is equivalent to the 6 ATP. So with the help of this we can gain the 8 ATPs. Directly we can gain only the 2 ATPs, but with the help of dehydrogenase of that coenzyme we can gain the 8 ATPs. So if the question is how many ATPs we gain in glycolysis, that is your 8 ATPs. We synthesize 4 ATPs. Along with that we are having the NADH and 2 are being utilized. Total 10 में से 2 माइनस किया तो अल्टीमेटली गेन कितना हो गया? नेट गेन इज 8 ATPs. Fine. So this is very important that you have to learn the systematic representation of glycolysis. What is glycolysis? The breakdown of glucose 6-carbon compound into pyruvic acid, that is your 3-carbon compound with 2 molecules. Fine. And while these steps we are having the 10 steps, that is a systematic formation of 10 steps. And if your substrate is sucrose instead of glucose, so what will happen? So sucrose, that is your disaccharide, first it has to be converted into glucose and fructose with the help of enzyme invertase, and then your glycolysis will going to start. So again this is a very important topic. अगर substrate आपका glucose की जगह sucrose होगा तो पहले sucrose glucose में convert होगा with the help of enzyme invertase, and then the reaction will follow same as that of the glycolysis with glucose. Site क्या है? That is your cytoplasm, and it is a common pathway for both the aerobic and anaerobic. या कोई भी respiration अगर शुरू होनी है body में तो सबसे पहले जो ग्लूकोज हमारा रेस्पिरेटरी सबस्ट्रेट है, इट विल अंडरगोज द ग्लाइकोलिसिस, दैट इज pyruvic acid, देन डिपेंड कि whether it will go to mitochondria in the presence of oxygen or it will without the oxygen, they will form the ethyl alcohol. So please go through this, note down, and for the next session we will discuss about the Krebs cycle as well as the electron transport chain in the aerobic respiration and fermentation in the anaerobic respiration. Till then, thank you, have a nice day.